So like Emily said, my presentation is on potentiating ALS breakthroughs. And this is one of those words. The communications team at TDI teases me about my favorite words, and this is one of them because I think it's the right word. Potentiating. It's to make something more likely to happen. And so we do want to make breakthroughs more likely, and we want to make breakthroughs that are on the cusp more likely to come to fruition. And so that's what we aim to do at ALS TDI. So ALS, I think everybody here is very familiar with what ALS is, but I'm going to talk a little bit about it to provide just a framework for the challenges that we face. So ALS is a neurodegenerative disorder that primarily affects upper and lower motor neurons. So the upper motor neurons are in the brain, lower motor neurons are in the spinal cord and connect out to the muscle. And in terms of ALS, that's essentially the most that everyone who develops the disease ends up having in common. ALS can really manifest many, many different ways, particularly in terms of its onset. So some people can have primary symptom manifestation secondary to dysfunction of the motor neurons that are in the brain. Some can be focused on the ones in the spinal cord. Some can manifest with symptoms affecting their limbs first or their ability to speak and swallow, so bulbar onset. There are many, many different clinical manifestations of the disease, and even some people with ALS develop cognitive or behavioral impairment as well. It's a very heterogeneous disorder. That's a term that you're going to hear a lot today. It's also heterogeneous in terms of its underlying genetics and biological processes that are engaged. And we've learned a lot about that when we study familial ALS, so ALS that's directly inherited from a parent or a grandparent to a child or grandchild. And we see many, many different genes which, when mutated, can end up increasing the likelihood of developing this disease. And so we understand ALS now as really more than one condition, which means it's going to take more than one approach to develop treatments. And so sort of downstream of that, there are a number of challenges that we confront when we're trying to develop treatments for the disease. What I just said, it's not one single disease, and it may manifest a lot of different ways. Another challenge that we confront, and more people with ALS confront it more starkly than we as researchers do, is that the ALS clock is not necessarily synced to the drug development. Development of treatments for any disease can take 10 to 15 years, and that's really an unacceptable outcome for people who are diagnosed with a disease like ALS. One of the other challenges that we've confronted is that it's actually been very difficult to measure when a therapeutic is being effective and in whom it is being effective for ALS. So that's a gap that we need to address. Another challenge sort of downstream of that is that these different endpoints that we do use to measure effects require us to use long and very large studies. So it takes a lot of resources and a lot of patient time and a lot of clinic time to really determine if something is being helpful. And trying to address that, as we're recognizing that ALS is a heterogeneous disorder, trying to find the right people for the right treatment or therapeutic approach remains a big challenge that we're confronting right now as a field, and we at ALS TDI are trying to confront. And so we actually are pretty well positioned as an independent nonprofit biotech company to try to address that. We get to prioritize our efforts rooted in making an impact for people with ALS, not chasing after any profits. And we get to think about both the short term, right, what can really be effective for somebody dealing with the disease right now, while also taking a long view on what can be really effective for the entire field to kind of raise all boats, recognizing that we're not going to be able to get this done alone, right? The entire ALS research field is going to be necessary to deliver the treatments to all of the different people affected in the most expeditious manner possible. So we think about that, how we're influencing the ecosystem, and I'll tell you more about that today. And so our programs, we certainly have internal research programs, but we also have a lot of collaborative programs where we bring ideas from folks from academia, from industry into our lab and really try to expedite the bringing those projects to fruition. So Emily talked a little bit about ALS TDI. We were founded in 1999 by a family that was directly affected by ALS, the Haywood family, when Stephen Haywood was diagnosed with ALS at the age of 29. He's the young man in the orange t-shirt back there. And so we are a brick and mortar lab located about 5 miles from here in Watertown, Massachusetts, with about 30 multidisciplinary researchers focused on ALS. And really we have two major objectives. One is to invent and advance treatments for ALS as quickly as we can, doing preclinical testing in the lab. But we also have this other objective, which is to contribute to the creation of an ALS research ecosystem where drug development and drug discovery is easier and faster. As far as our team goes, the research team really breaks into three big categories. We have a cell biology team that takes advantage of induced pluripotent stem cells from people with ALS. So you collect cells, either skin cells or blood cells, and you can turn them into stem cells, which can then be turned into things like motor neurons that give you the opportunity to study the biology and to test drugs. We also have a world-class preclinical pharmacology team working with animal models of ALS. That's been kind of cornerstone of our research program from the very beginning back in 2000, 2001, where we've did a lot of work with SOD1 mice, but actually expanded into multiple models in the past few years. And then all of that connects and interfaces with our clinical translational research team, which oversees the ARC study, which you'll hear a lot more about from both Alan Primasiri later this morning and Dr. Danielle Boyce. So this is a framework. My brain often thinks in graphs. I'm weird. So this is a framework that I use to think about our opportunities in target discovery and drug discovery for ALS. And we talked about how it's likely not going to be one therapeutic that'll ultimately address the needs of all people with ALS. If it was, this is what it would look like. This entire graph would be filled. All of the people with ALS would be completely rescued from the treatment. So the y-axis is the drug efficacy at the top of the bar, and all of the people on the far right of the x-axis. That's very unlikely to be the case. It's going to take multiple approaches to really make a difference. You may end up having a therapeutic that looks a little bit like Riluzole, where maybe the efficacy is not profound, but you may have many, many responders to that. You may have a drug that looks more like tofersen, like this, where you're seeing very, very high efficacy, but in very, very few patients. In the case of tofersen, it's SOD1 mutant carriers. So in fact, I had the privilege of being at the NEALS meeting last week, where Dr. Tim Miller announced that of SOD1 carriers who take tofersen, about 25% either completely stabilize or get better, which is incredible, right? The idea 5 years ago that a person with ALS could get better was not something that we often let ourselves imagine, and we can imagine that today. But then you can imagine other therapeutic approaches, maybe a treatment that influences the immune system and maybe benefits about half of the people with ALS, more than Riluzole, but still not completely filling that gap. And then you can think about something that helps the rest of the folks with SOD1 mutations. And now maybe an ASO against other ultra-rare mutations starts to treat others. So you can see what I'm trying to get at is ultimately we need to fill this whole box. We need to fill this box for treatments for everybody with the disease, but it's going to take a lot, and it's going to take the entire village, this entire community, and we want to help make that happen. But how do we think about the therapeutic approaches that we're bringing into our lab? And that's something I wanted to talk a little bit about today. So again, I think in graphs and figures. The ideal drug would be one that has that transformative impact. So it reaches out in this radar plot to the very edge. It's going to have the biggest impact. It's going to, treat somebody completely, maybe even cure them. The ideal drug would be one that has potential to do so for everyone, all subtypes, so the very far edge of that radar plot. And it would be one where we know a lot about the underlying biology, so we would have really, really solid position to do the project. And in the last category, no one else would be working on it. So we wouldn't be having any redundant effort. This doesn't exist. This is the ideal project, and nothing like this actually exists. So there are always trade-offs when we're thinking about the projects that we're bringing in-house, and this is kind of like a hypothetical opportunity here. But I'm actually going to dive into some of our specific case studies and the way we've been thinking about them. And so one of them is a drug called TDI 1831. This is a molecule that we've been working on for about 5 years, and we actually came to study this molecule thinking about it more in terms of general effects on mitochondrial biology, oxidative stress, and general biological energetics. But in recent years, as we were working on this project, findings emerged. There was a case study published, I think, by Dr. Bob Bowser's group and a couple other genome-wide association studies that identified mutations in a gene called ATP7A, which is actually responsible for another pediatric neurodegenerative condition called Menkes disease, where some people with ALS, ultra-rare cases, seem to have these mutations. This is not a consensus gene yet. This is not one where we have Mendelial evidence that it causes ALS, but it's one that we think is potentially critical. And we looked in our own ARC study, and we found instances of these mutations in our data set. And so we started testing this drug in models of ATP-mediated neurodegeneration and really saw pretty profound efficacy from that. And so in this case, we're looking at a project where the subtype reach might be actually quite low, but the potential for impact might be really, really high. We might really, really be able to help these folks with these very, very rare mutations. There aren't a lot of folks working on this project, so it's quite unique. But our understanding remains sort of intermediate to low, right? We don't know a lot about these specific mutations in the context of ALS. We know a lot about it in the context of Menkes, but not ALS. So, but it's still a project that we're really excited to move forward. Another project that I wanted to talk about that maybe you've heard about, it's a target space that we've been exploring with small molecule drugs. They're called PRMT inhibitors. They're protein arginine methyltransferase inhibitors, and you don't have to remember that, or you could watch it on the video later. But we came to this by way of looking at the implications of toxic dipeptide repeat proteins that are formed in cases of C9orf72 mediated ALS. So C9orf ALS is a really common mutation in familial ALS. It even shows up in some sporadic cases. The downstream biology is really complex, but one of the areas that seems important is production of these toxic proteins. And so we looked at what might be important interactors with these toxic proteins, and we found that inhibitors of these enzymes were actually really, really efficacious in cell-based models of ALS. And so we began working on those a few years ago. We think that the potential for impact in C9 is potentially moderate, or the high end, where you're going relatively high up in the biology and you may be able to have a pretty big impact. Our understanding of this is growing because we've been working on it for some time. We've even recruited some help from folks who are in the building here, collaborators of ours at Einstein School of Medicine. Eliza Silverstein has a poster she'll be presenting on this later. And this is really a quite unique project. Not a lot of folks are working on this right now in the ALS space, so we believe we're filling a gap. As we've thought more about the biology here, we also think about it outside of the context of C9orf72 ALS. We're thinking about this also in the context of sporadic ALS, because arginine methylation is really, really important in modulating RNA metabolism, splicing, and stress responses, all things that have been implicated in both familial and sporadic ALS. So you can see this sort of green radar plot sitting on top, where maybe the subtype reach could be much, much broader. Maybe the impact, because it's a little bit further downstream, might be less. And we know a whole lot less about it in this context right now, but it still remains a very unique project that we're focused on. And then the last sort of case example that I wanted to bring up today are lipid nanoparticles for developing delivery of mRNAs as a therapeutic for central nervous system diseases like ALS. So in ALS, we're learning more and more that there are some proteins and some RNAs that are underexpressed, that you don't have enough of them there. There are a lot of conditions a lot of ways to think about targets where we're pretty good at reducing the amount of something. We aren't as good, at least in the brain, of figuring out ways to increase the amount of something that might be in deficit. And so we've been working hard with a collaborator. I think Mike Mitchell is here somewhere. We've been working with Dr. Mitchell at UPenn to really develop an approach that would deliver these mRNAs directly into the brain and spinal cord to replenish proteins that are in deficit, and we're excited about that. So the subtype reach could be really, really broad because this is a technology platform that allows you to go after many, many different targets. The impact on disease could also be really, really profound if you found the right specific target. This is quite unique because I don't think there are a lot of folks working on getting mRNA LNPs into the brain and spinal cord, particularly for ALS. But our understanding is not that strong yet, though we do have the crutch of working with an expert in the space at UPenn, and so we're making strides there. But we do more than target discovery and therapeutic development at ALS TDI. Again, we're also trying to be sort of an ecosystem enhancer. And so you'll hear again from Alan and Danielle about the ARC study, this large natural history study and omics study that we're collecting a lot of data from people with ALS, learning about the disease from people with the disease. And our model for the past few years has really been to get the data out there. We certainly use the data ourselves, but we want to sort of democratize the information to get them to others in the field who can really do a lot with it. And so we've been opening the data through the ARC Data Commons, which any academic or nonprofit can get access to with no cost. There's a small fee if you're a for-profit company. We also have been collecting a lot of samples, and those samples are also available for targeted studies, looking for biomarkers or even using cells to do cell biology studies and look for target discovery, target advancement. And then we also leverage our own expertise in-house. Kyle Denton, our director of cell biology, does a lot of this, working with collaborators like our friends at Corsilex, to conduct therapeutic screens and cell-based models of ALS. We work with a lot of different folks this way. And then our preclinical pharmacology team often tests a lot of drugs in animal models of ALS for a lot of different folks. So our resources are the ALS field's resources. Speaking to that, a lot of key collaborations in 2025 that I kind of wanted to highlight. This is not a comprehensive list, but a few that I wanted to speak about here. We're excited to have partnered with Augie's Quest and Answer ALS specifically this year. We're going to launch sort of a complementary study to the ARC study that I've just described that's going to be focused on identifying and understanding the increased risk of ALS in veterans and elite athletes. We think that there may be an importance in understanding those subsets, and it might teach us something about the biology. And so we'll be launching the Champion Insight study, which ultimately the analyses we conduct in that study will bleed over into the entire ARC study to really give us a really comprehensive understanding of the metabolomics, so sort of the bioenergetics of ALS. And we're grateful to work with Augie's Quest and Answer on that. We're also working right now with Critical Path Institute, CPath, to really, and actually MGH investigators, to make the most sense of the accelerometer data that we've been collecting. We send these devices to folks in our ARC study where we can track the ability to move your arms and legs. And to those of you at home or folks here who contribute those data, thank you. We're working really, really hard with CPath, an entire sort of industry and academic coalition to try to make the best use of these data to find the specific metrics that could be measured in a clinical trial to see if a drug is working. Down on the bottom left, I want to highlight a collaboration with a company in the UK called Axol, where we've taken cells that we've collected from participants in our ARC study to turn them into induced pluripotent stem cells that can now be motor neurons that anyone, any company, any academic lab in the world will be able to access through Axol. They'll manufacture them at scale, so we don't have to become a manufacturing company. That's not what we want to be, right? But they'll be able to study sporadic ALS in those cell resources. And then I mentioned earlier CourseLex. I want to thank them for their partnership over the past year working on C9orf72 focused therapeutics. But with us here today, I've already called some of them out, our collaborators from UPenn, Dr. Mike Mitchell, Dr. David Schechter from Einstein, our folks running the Longitude Prize, who you'll hear from today, Tris and Amalia, our collaborators at LifeArc, also in the UK, focused on biomarkers and cell biology. And then you'll hear on the panel today from Arena, from Trace Neuroscience, a fantastic new company developing an ASO targeting UNC13, and our longtime collaborators at EcoNeso as well. So excited to welcome them all here today. So in short, we're trying to work hard to contribute to this healthy ALS ecosystem. We think we're uniquely positioned to really interact directly with the patient community as a nonprofit. We work well with other nonprofit organizations to leverage our complementary strengths. We can bring in resources from investors and philanthropists, and we're increasingly working with regulators to work on digital biomarkers and our longtime collaborators in academia and biotech. Thank you to all the people with ALS and their loved ones who have been a part of this entire effort through the past 2 decades. The team at ALS TDI is incredible. It's an honor to work with you and all of our collaborators and our financial supporters. Thank you.
Thank you, Fernando. We'll now open the floor for questions. For those of you who are here in the room, you can go ahead and raise your hands if you have a question, and we will bring you the microphone. If you can identify yourself and your connection to ALS, that would be great to give context to folks online. And if you're joining us online, feel free to type in your question, your name, and your connection in the chat, and we will get to those questions as well.
Cool.
Thank you. That was very interesting. Your graph, where you look at different approaches to different variants and so on. I'm wondering whether you're looking at, or what you see the potential being of combination therapies and approaches where drugs are tackling different things at once, and if we should be looking at that, at the iPSC stage when we're starting to screen potential molecules.
Yeah, it's a great question, and one that we actually do think about a lot. It increases the complexity of the study. It's an exponential increase in complexity, and when the experiments are already really, really large and unwieldy, sometimes there's a trade-off in the interpretability of those types of experiments. So we revisit it constantly, like this question, both at the cell-based level and also in the in vivo pharmacology level, right? It's not just an instance where you can just pick a dose and stack the same, an arbitrary dose of another. You end up having to do dose ranges on dose ranges. And because there are some concentrations or exposures of a drug that can be complementary and end up being additive, where you see a bigger effect, but they can also be antagonistic. And so at the end of the day, if you don't see an effect, you wonder, well, did you get in the way of a drug effect by having the other drug there? So we do think about it, and we have done those types of studies, but it's usually with molecules that we already feel quite strong about their ability to be helpful independently. Thanks for that question.
Mike Mitchell, University of Pennsylvania. We collaborate with Fernando and his team on a few research projects. So thank you so much, Fernando. That was a great opening address. I learned a lot. In terms of the therapeutics that you mentioned, I was curious, is the field also thinking about therapies in terms of autoimmunity? This paper that came out in Nature a couple of weeks ago, I think it was La Jolla Institute of Immunology, they were looking at the autoimmune response to C9orf. And I was wondering, is the field as a whole, is anyone thinking about this in terms of autoimmunity, in terms of a therapy?
The immune system has been implicated for actually a long time in the ALS space. So, gosh, it was actually Dr. Stan Appel, probably going back to the mid-90s or maybe even earlier, who really was bringing a point of emphasis that the immune system matters. And we agree that it does matter. In fact, one of the drugs that we invented that moved into trial and is a little bit stalled in phase 2, going to phase 3, is targeting CD40 ligand, right? It's one of the key regulators of the immune system in the context of ALS. So we think it's likely important, maybe not the same degree of important in everyone. And so one of the key elements that we're trying to think about right now is, well, what role does it play? It seems like when we look in our samples, we seem to see that faster progressors do have more engagement of the immune system, or dysregulated engagement of the immune system. But it doesn't always look the same way. And that might mean that there are phases where it looks like one thing and then it looks like another thing. And so there's a lot that we still need to understand, but we certainly do care about it. The C9orf72 question specifically is new, right? And so looking at that subset and thinking about how either dysregulation by gain of function or loss of function of normal C9orf72 could increase likelihood of an autoimmune component is something that does need to be explored specifically. Thanks for bringing it up. And good to see you in 3D. Yes. Yeah, that's a question here. Oh, there too. Okay, sorry.
Hi, Fernando. I know you, and so you might kill me for asking this question, but given the different sort of case studies that you talked about up there, and also mention of the ALS clock, and we all know that science is the opposite end of that, so it's going slower, but what would you say is the thing you're most excited about in the lab, if you had to kind of putting into context the timing that we're all, you know, if it would have happened yesterday, it wouldn't have been soon enough. So I guess I'm curious, like, what do you feel is kind of furthest along, most excited about?
The closest to people, though very few of them, is the copper complex, right? So we're in the middle of IND-enabling studies for that small molecule drug. We will then ultimately be working with clinical investigators to try to identify the very rare instances of folks who are carriers, and we would want to try the drug in those folks. So that would be sort of, when I'm thinking about, you know, what's the closest, that would be it. I was in the lab, I was telling somebody this yesterday at the open house, we kind of are in a privileged spot in a way, where certainly we are experiencing really viscerally the ALS clock because we know so many people with the disease and we're seeing the progression and it hurts. But not as much as it does for the person with the disease, right? And so when that happens, you get to kind of bounce into different time frames of contemplation, right? So I get to, when I get really, really uncomfortable, I can hop to thinking about the future where there are a lot of treatments that are there because I know the work that we're getting done and others are getting done that'll get there. And I can also hop into a data set that, you know, Cindy Gill sends me 2 days ago that lifts me up, that there's a project that we're doing that is moving really, really quickly. But then you do always have to revisit at the start of the day and the end of the day the ALS clock. And it's a reality that we have to deal with. But we're mindful of there are a lot of other folks who are dealing with it in a much harder way. I don't know if I answered your question, but.
Hi, this is one online question from Lynn.
There was a question here from Carmen too, so sorry.
Yes, hello, I'm Carmen Mikolajczyk. My husband was Tom Mikolajczyk, who dedicated the last years of his life to obtaining recognition of veterans as having service-connected disabilities, at 100% of them. Mine is sort of a two-part question. Have you had any collaboration with the veterans, the project they had where they obtained brain and spinal cord for study? Because we did that. He donated his brain and spinal cord. And I'm wondering if any preserved tissue there could benefit your research at this stage of time, at this stage of research, and how we might get that. Like, for instance, I would be able to say that I would want that shared with you, if that is a possibility, if it has any benefits that in your mind.
Yeah, there are great biobanks that have been created in the past few years that make those types of samples available. So I know the VA, the CDC, and Target ALS are really great resources for postmortem tissue at this point. And so we recently actually submitted requests for those types of tissues in the past year, and we're starting to see those come in. It's kind of like an onerous, like, bureaucratic process, but it does work. Like, it does end up to get to us. So thank you to you and to your late husband for contributing those samples, because they do matter. So.
And also, it just brought up that when I received a report some months later from the VA, one of the comorbidities they identified was Parkinson's.
That's really interesting. So when you, I recently looked up, because I actually got to know a person who had both ALS coincident with Parkinson's in the past year. And I looked it up. Those two conditions specifically are really rarely described happening at the same time in studies. So I'm curious to hear about that report. Yeah.
So we could possibly request that specific report. I mean, I have a copy of it, so you can
I'd love to review it,
for
please.
Carol. Okay.
Yeah. Thank
great.
you.
Thank you so much for all you're doing.
Thank you. And I know we had one question.
Yeah, we have one more question.
And sorry, I'm
online.
sure I'm way over
No, you're
time.
fine.
Yeah.
So this question asked, do you use a similar process as the DoD CDMRP ALSRP program to winnow ideas into funding?
No, to be short, that's like a really, so the CDMRP, ALSRP grant review process is kind of a collection of, we submit grants to this funding mechanism all the time. It's a little bit like the NIH. Many groups do. And so there will be, somewhere between 200 and 400 applications exploring really fleshed out projects in order to get funding for the project. And that ends up requiring bringing in hundreds of reviewers that help kind of vet through these. And they're really looking at fully baked project ideas. That is not the process that we go through. We have a team of 30 scientists who are collaborating informally or formally with lots of other folks. And it's a much more free-flowing sort of real-time evaluation of ideas rather than looking at a project that somebody, I'm getting told that there's time's up. So in short, it's the same rigorous review of science, but not always looking at a fully formed idea or fully formed project. So I hope that answers the question. Thank you all.